evening, everyone. Welcome to my YouTube channel today. We'll be talking about urine proteins. I hope you enjoyed the video. So in our previous videos, we talked about plasma proteins. You can go and check that one out. But before you do, you make sure you hit on the subscribe button. You can also like and share. So pretty quick, let's go through urine proteins. It's going to be a short lecture, but then a vivid one as well. So we are going to go through the reason why the kidney cannot filter protein, abnormal proteinuria and symptoms. Then we'll go through the possible causes of proteinuria. Then we discuss types of proteinuria. Then we'll talk about nephrotic syndrome as our case study. Then we'll talk about testing for urinary proteins. Then finally we'll discuss Ben-Jones proteins and multiple myeloma. Alright. So pretty quick. So the first question we want to ask ourselves is that why can't the kidney filter proteins? We all know that it is the nephron, that's the basic unit of the kidney, that is responsible for the filtration. And within the nephron, it is the glomerula that is responsible for the filtration of most substances. So the question we should ask ourselves is that why can't the kidney filter proteins? Now, the kidney has a renal threshold, or a particular size of molecule that it is designed to filter. So anything less than 79 kilodalton, the kidney is unable to filter. Anything greater than 70 kilodalton, the kidney is unable to filter. Now when we look at the plasma proteins, we can have albumin. Albumin is approximately 69 kilodalton, which is almost 70 kilodalton. So the kidney is unable to filter albumin. And then most of the other alpha and beta proteins, they have a larger molecular size, which makes the kidney unable to filter. So the kidney has fenestrated endothelium. When we say fenestrated endothelium that are so tiny to allow certain molecules to be filtered. So that is the main reason why the kidney cannot filter proteins. Therefore, if there is protein found in the urine, it means there is an abnormality in the kidney. So it could be as a result of a glomerulus abnormality. It could be as a result of the renal tubule abnormality, etc. So that's the main reason why the kidneys cannot filter protein. Now, abnormal proteinuria and its symptoms. Abnormal, we can say that a person has an abnormal proteinuria when a person excretes about 150 milligram and above of protein per day. So when there is about 150 milligrams of protein per day, then we can say that the person has an abnormal proteinuria. Now, proteinuria itself do not have a specific symptoms, so no specific symptoms. Proteinuria do not have no specific symptoms, but then such urines have a frothy appearance and are mostly likely to be associated with symptoms like edema. So edema. So we know that once there is a loss in protein, if you haven't watched our plasma protein videos, you have to go and watch. You realize that when there's a large loss, or when there's hypoalbuminemia, when there's a low albumin as a result of large excretion of proteins through the urine, there can be what we call edema. So that can also be a symptom of proteinuria as well. Now, let's go to the possible causes of proteinuria. What do you think will be the possible cause of proteinuria? So first of all, we have diseases that affect the glomeruli. So we have diseases like glomerulonephritis, glomerulonephritis. Then we also have diabetes too as well. Then we can have urinary tract infections. Then we can also have symptoms of other diseases such as eclampsia in pregnancy. So eclampsia in pregnancy. Then we have congestive heart failure. So glomerulonephritis, when there is an inflammation in the glomerulus, it can cause protein to leak into the urine. And when there is also urinary tract infection too, that can also cause that. And we have symptoms of other diseases such as eclampsia in pregnancy and then congestive heart failure. So these are the ways that the, let's say, pathological ways that we can have protein in the urine. Now when we come to physical ways or other external factors that can affect protein in urine, early morning urines too. So it is known that early morning urine contains a lot of protein. So when you want to measure proteins, it's not advisable to take early morning urine. You want to measure urine protein. Then also after vigorous exercise, there's some amount, significant amount of protein in the urine, and this too can falsify results, especially when you want to find proteins in urine. Now, there are basically three types of proteinuria. So we have the glomerular proteinuria, we have tubular proteinuria, and we have postrenal proteinuria. So the glomerular proteinuria is actually the filtration of macromolecules such as albumin. So when we talk about the glomerular proteinuria, we are talking about the nephrons glomerulus. So when there is a damage to this glomerulus, there is filtration of macromolecules like albumin. There is a filtration of macromolecules like albumin through the glomerulus into the renal tubules. Uh, so this is one cause of proteinuria, glomerular proteinuria. Then we also have tubular proteinuria. So the tubular proteinuria is also a excretion of low molecular weight proteins such as the beta globulins. So this tubular, when you talk about urine formation, there's a stage we call tubular excretion, and this tubular excretion, this is where the renal tubules excrete certain substances into the urine, and some of these substances can be the beta globulins, which might be found within the urine. So it's not always that the urine that is found in the urine is albumin, but mostly it's albumin that is what related to proteinuria. But sometimes we can have other proteins such as the beta globulins too, or the beta proteins. Then we have the postrenal proteinuria. So this one is as a result of inflammation of the urinary tract. So we can talk about inflammation of the urinary tract. Then we can talk about urinary tract infections as well. Okay, so that is it for the types of protein. So you have to bear in mind that we have glomerular proteinuria, we have the tubular proteinuria, and we have the postrenal proteinuria. So this one happens in the glomerulus of the nephron. This one happens in the renal tubules where there's a excretion of protein molecules into the filtrates. And we have the postrenal proteinuria, which is caused by inflammation of the urinary tract and urinary tract infections too as well. All this can result into the excretion of protein into the urine, or distribution of protein into the urine. Now, our case study. Let's talk about nephrotic syndrome. So, nephrotic syndrome is basically a disease that is characterized by one, massive proteinuria. Massive proteinuria. So, it is estimated that at least 3.5 grams per day. So, this is a massive relief of proteinuria, and this can lead to symptoms such as hypoalbuminemia. So there's low what plasma albumin, and this would definitely lead to generalized what edema. Generalized edema. Generalized edema. So this is one. This is what the nephrotic syndrome is about. So when you realize that there is a massive protein release, and then we measure low plasma albumin, which has a physical manifestation of what generalized edema, then we can say that the person is suffering from what? For nephrotic syndrome. Okay. So one characteristics of nephrotic syndrome is that there is hyperlipidemia. So when there is a low plasma albumin, the body's compensative response is to overproduce lipids. So as a result, there is hyperlipidemia. So if you really want to diagnose nephrotic syndrome, what you have to look for is liver function test. So in the liver function test, you have to take notice of the total protein, and then especially albumin. Then you are going to also do lipid profile. So when you realize that there is an increase in, let's say, LDL or triglyceride, together with a drop in what total protein or albumin, and also the presence of what albumin in urine, then we can diagnose what nephrotic syndrome. Alright, so that is one thing you have to take notice of. So anytime you see nephrotic syndrome, what you have to look out for is a drop in what plasma albumin and total protein, the presence of what urine protein, and also an elevated what LDL or triglyceride in lipid profile. Alright, so we have various tests that we can use to determine what protein in urine. In fact, determining protein in urine or urine proteins is not any big deal, because it doesn't require any high technical skills to determine what protein in urine. So first of all, most of these tests are qualitative. It's only a few that are quantitative. So this qualitative, it either gives a color indication or something, a positive negative kind of thing, for you to determine whether there is a presence or absence, but it doesn't give what a quantitative measurement of the amount of protein that is found in the urine. So for instance, there's a method called Albutex method. Albutex method is an old method that is used to also determine what the presence of protein in urine. It's just like the dipstick method, but this one, it requires sophisticated reagents and urine samples, etc. But now, in our routine laboratory, we use the dipstick method. So the dipstick method actually has specific reagents indicated in the dipstick. So for instance, there's a column in the dipsticks that is also only responsible for for detection of what proteins. So once we dip this column into the urine, there's a reaction between the reagent and then the albumin content. Mostly is the albumin content of the urine. So once the reaction occurs, once albumin reacts with the protein, the reagent, there's a complex that is formed, and this complex can show a color indication, and the color indication will determine the presence of what of of protein. Then we have the SSA method, sulfosalicylic acid tests. That one is also a method for determining protein. Mostly it's not used. Then we have the turbidimetric test. That one we check the turbidity of the urine and compare it to a standard to determine the presence of what? Of urine protein or albumin. Then we have ELISA, and then electrophoresis as well. Mostly these two are used in research. Alright. So these are the basic method of testing for protein in urine. Now let's talk about multiple myeloma. So multiple myeloma is basically the abnormal proliferation of the immune cells, one or two of the immune cells, especially the B cells or the plasma cells, over proliferation of the plasma cell. So it's actually a bone. It's also known as the bone marrow cancer or multiple myeloma, and it is characterized by easily breaking of bones, confusion, dehydration, excessive thirst, and then frequent urination. So these are the symptoms that we have, and this multiple myeloma leads to the release of a protein we call the Benz-Jones protein, which can be detected in the urine as an indicative of what? Multiple myeloma. So there are specific tests that we use to detect Benz-Jones protein in the urine. So we have the hydrochloric acid test. Hydrochloric acid test is a type of test that we can use to determine the presence of what? Benz-Jones protein in the urine. So that will be it for multiple myeloma. So basically this is a brief overview of urine proteins. In the next video we'll talk about cerebrospinal fluid proteins, and then we'll go much deeper into that. Thank you very much for joining today's session. Have a wonderful evening.